So far in this section, we've mostly focused on various pathways to fix carbon dioxide. But once you've got those precursor molecules, whether they're formed by autotrophs or consumed by heterotrophs, you then need to use those to actually produce the things that are used in bacterial cells. So things like monosaccharides, disaccharides, things making up the peptidoglycan in the cell wall or even the cell membrane, amino acids, and lipids are all some of the key things that cells have to produce. And so from these compounds, you can basically arrange them into really straightforward ways, just with these carbon, hydrogen, oxygen molecules. Other compounds, especially like amino acids, need to be produced by more complex pathways and or by modifications to these molecules. So when we talk about amino acids, one of the key things that makes them kind of special is that you also need to incorporate a lot of nitrogen and sulfur into some of them, making the process of assimilation or synthesis a lot more complex. If we're talking about more straightforward things, one of the easiest ones to think about is gluconeogenesis, which is the generation of glucose, and it's mostly just a reversal of the glycolysis pathway. And so for the most part, the enzymes will be shared, although there are a few that are completely unique to gluconeogenesis, but you're basically going in reverse. So remember, glycolytic pathways, including glycolysis or the EMP pathway, go from glucose to pyruvate. Gluconeogenesis will basically reverse this, going from pyruvate to glucose. Alright, so that one's pretty straightforward. Another really important component of the cells are fatty acids, which have to be synthesized in this complex called the fatty acid synthase complex. And so this contains the enzymes necessary to basically generate the fatty acid from acetyl-CoA. And there are a lot of variations that can happen as a result within this complex, including variations on how saturated the membrane or the fatty acids will be, depending on the temperature in the cell at that moment. So this can be really complex. We're not going to look at it all that closely. But basically the gist is that through a series of pathways, you take your acetyl-CoA that gets modified, and you can basically add additional CH2 molecules to continue reducing, and you can reduce that and build basically a fatty acid of indefinite length. Then that will be generated as a saturated fatty acid, but you can make modifications to have that be an unsaturated fatty acid if the cell is experiencing like colder temperature or higher pressure. Another kind of special one that's again following the same basic component is the bacterial cell wall. And so this follows that same idea of forming these precursors and combining them into special ways. And remember, this is really important for the bacterial cell, and this is one of the main targets of antibiotics. But what happens is the precursors get formed inside the cytosol, and then they're just shipped out where they're processed by enzymes outside in the periplasm. Alright, and so as I was saying, amino acids get a little bit more complex, and before we can actually look at how those are synthesized, we need to talk about how nitrogen is either fixed or nitrogen and sulfur can be assimilated. So we'll kind of detour, put a pause on biosynthesis for now, and stop just with this conversation of carbon.